Human tuberculosis is the leading infectious cause of death globally, resulting in over 1 million fatalities annually. It's especially prevalent in India and some countries in Africa, since the treatment for tuberculosis consists of around 6 months of 4 different costly antibiotics. Also, there isn't an effective vaccine. This means that there is nothing to prevent infection, and when someone's infected, they must take a long course of highly expensive treatment. However, recent research has revealed a link between a bacterium found in a sponge, the Mycobacterium Spongiae, and tuberculosis that may provide more insight into curating a vaccine. The origin of the Mycobacterium Spongiae and its relation to M. tuberculosis is quite perplexing. Researchers at the Peter Doherty Institute for Infection and Immunity were initially collecting samples of bacteria in Cooktown, Queensland. By chance, they collected a sample of the sponge and later analyzed its genetic properties, which led to the shocking discovery of its shared genome with M. tuberculosis. Looking into the origins of M. tuberculosis provides evidence that marine mycobacteria may be ancestors of the M. tuberculosis bacterium. Through various types of analyses, M. spongiae was shown to retain many of the virulence factors that M. tuberculosis contains, such as intact secretion systems. Intact secretion systems are complete cellular structures that mediate the transport of molecules, commonly proteins, from within the cell to outside the environment or inside other cells. However, despite these shared virulence factors, M. spongiae does not remain pathogenic in animal models. This seemed to be a breakthrough in the search for a vaccine, but M. spongiae did not provide protection against M. tuberculosis when used in infected mice. While it is disappointing that M. spongiae ended up not being the solution to the vaccination issue of tuberculosis, it did provide more insight into the evolutionary trajectory of M. tuberculosis. The figure shown to the right is a figure from a paper authored by Dr. Sasha Pidot and his colleagues. It depicts a phylogenetic tree with 1,000 bootstrap iterations. Bootstrap support is a statistical method used in phylogenetic analysis. This phylogenetic tree is the maximum likelihood phylogenetic tree that shows the evolutionary relationships of 30 Mycobacterium species. An asterisk on any particular node reflects a bootstrap support for that branch greater than 60%, meaning that these branches have less than 40% uncertainty. All the remaining nodes have a bootstrap support of greater than 90%, which reflects high confidence in the majority of branching patterns.
M. spongiae is important because of its shared similarity and character with M. tuberculosis that was essential in understanding the evolution of TB and its pathogenesis. Despite M. spongiae itself being ineffective as a vaccine, studying it can help us develop new strategies in protecting against TB and lead to developing other potential vaccines. The research done by Dr. Sasha Pidot and his colleagues deals with the multitude of concepts that we have learned about in biology thus far, especially evolution. The evolutionary trajectory of M. spongiae resulted in a concept within evolution called phylogenetic trees. Earlier we talked about the phylogenetic trees created by the researchers that show the ancestry of M. tuberculosis. Phylogenetic trees show that hypothetical evolutionary relatedness. One of the conclusions within the paper was that M. tuberculosis likely evolved from a marine bacterium due to the close phylogenetic position of the M. spongiae. Dr. Pidot's work also shows that the ancestor of tuberculosis-causing mycobacteria had an unusual capacity for adaptation to specific environments. This deals with the concept of adaptation within evolution, in which adaptation is the process by which a species becomes better fitted to its environment through natural selection. In conclusion, although the study of Mycobacterium spongiae did not yield a direct vaccine candidate, it has greatly contributed to the knowledge of the evolutionary origins of Mycobacterium tuberculosis. These genetic similarities provide insight into the mechanisms that allow M. tuberculosis to cause disease. This research indicates that studies regarding evolutionary biology and adaptation will someday provide innovative therapeutic strategies and better vaccines against tuberculosis, one of the most serious health concerns globally. Thank you for watching this video. We hope you learned something new about biology and the world you live in.